Hey, welcome back to today. I will discuss very briefly, just 5 to 6 minutes, the basics 3 of DNA, deoxyribonucleic acids. So, what is DNA and what is the single stranded DNA and double stranded DNA? How will interact this DNA with the ligands and how the DNA will form denaturation, how the DNA will damage. The basics 3 theories we will discuss today very briefly in the 5 to 6 minutes. We will move slide by slide. We can see the very basic things of what is DNA. So nucleic acids are the organic materials present in all organisms in the form of DNA and RNA. This point is very familiar to all that everyone knows. You can see the next one point. The nucleic acids are formed by the combination of nitrogenous bases like sugar molecules and phosphate groups that is linked by different bonded in series of sequence. So when you think the nucleic acid formation is basically the combination of nitrogenous with the sugar molecules and phosphate groups linkage and in the series of sequence. So that is called as a formation of DNA. The DNA structure defines the basic genetic makeup of our body. So DNA is the main thing is to carry the genetic information from one generation to another one generation. And is the part of, you can see here the next point, the part of the being responsible for the inheritance of genetic information in all living beings. DNA also plays a crucial roles in the production of proteins. So DNA is the one of the make the protein, and DNA is the one of the important part to formation of proteins to our body. And you can see DNA present in the mitochondrial of cells formed by the mitochondrial DNA. Suppose the DNA was formed in mitochondrial, then as usually we will call as a mitochondrial DNA. The DNA molecules consist of two long polypeptide chains composed of four. See, if how is formed the DNA? Suppose this is the one layer and this is the another one layer. See, forms like that one standard chain to another one chain means two polypeptide chains. So each one chains it consists of four types of nucleotides like subunits. So these chains are known as DNA chains, or simply we'll call the DNA strands. You can see here the hydrogen bonds are forming the portion of nucleotides hold to the two chains together. Suppose this is the one chain and this is the another one chain. So those chains, the interaction between these chains, formation of the making the distance by the hydrogen bond formation. So if you think each sugar block with the protering or knob, that is called 5-phosphate, and the other side of holes is called 3-hydroxyl group, means 3 ends. So it's complete chain. The formation of the interlocking the holes, moreover that ends each chain will be easily distinguishable. So you can see 3-hydroxyl group one side and 5-phosphate group is the one that simply will call tail of the each one stands. You can see here the three-dimensional structural of DNA is the double helix arise from the chemicals and the structural features of two polypeptide chains, as we earlier discussed this topic. And because of these two chains are held together by the hydrogen bond. Between these on the different strands on the basis of the inside the double helix and the sugar and phosphate backbones and the side chains outside will be A is always paired with T and G always paired with the C. What is A here? Adenine, the pair with the thymine, and guanine pair with the cytosine. I will show you the previous next slide in images. Now we can see actual what is that DNA, some more details. The DNA, deoxyribonucleic acid, is hereditary material to human almost all living organisms. Nearly every cells the person's body has the same DNA, but most of the DNA is located in the cell nucleus where the is called as nuclear DNA. Okay, that point is very important. So about the small amount of DNA can also be found in the mitochondria. So that is called as a mitochondrial DNA, or simply we'll call mtDNA. The information of DNA is stored in the code made up of the chemical bases, 4 chemical bases Like as you discussed the previous slide, the A means adenine, guanine, cytosine, thymine, T. These are the 4 base pairs of chemical base pairs is made up of one polypeptide chain, that is the one strands of DNA. Now you can move see here, the human DNA consists of 4 million base pairs. More than 99% of those base are the same in all the peoples. But the DNA is the double helix formed by the base pairs attached by the sugar and phosphate backbone. So DNA is the one of the long polymer made up of the repeating units of the called nucleotides. So when you discuss about the nucleic acid, this bit is very important. What is nucleotide? Nucleotides means is the one of the repeating units of made up of the DNA, that is called nucleotides. I will show you the next slide in image. The backbone of DNA based on the repeated pattern sugar groups and the phosphate group are the repeated in a sugar chain or the DNA chain, polypeptide chain. You can see here, this is the actual image of intercalation and grow binding and the electrostatic binding. You can see this image, you will understood what are the interactions will be formed in the DNA. Now we will move on to this DNA, one of the double helix and with the 2 strands connected by the hydrogen bonds as we discussed in the last slide. Now we can see both DNA and RNA consist of the major purine bases, adenine, guanine, cytosine, thymine, and the 2 major pyrimidine. So both DNA and RNA, one of the pyrimidine is the cytosines and second major is the pyridine is not in the both. So thymine in DNA and the uracil specially in is form in the RNA. Okay, the complementary base pairing enable to the base pairs with the packing with the energetically most favorable arrangement in the interior of the double helix. So each base pair of similar width and the holding the sugar phosphate backbones and the equal distance of the part along with the DNA molecule. Suppose this is the one DNA strand and this is the another one DNA strand. In between one strand to another one strand, there is a formation of hydrogen bond. These hydrogen bonds are have a equal distance. That point is very important. It have a equal distance of the combination of 2 strands of DNA. So the maximum of the efficacy of the base pairs packing in the 2 sugar phosphate backbones with around the each other than to form of double helix. So one turn like a, suppose this is the one turn to move like that. So one complete turn every 10 base pairs will form. Suppose this is the one turn. So this is having a 10 base pairs called as a one turn. So we will see that image, then easily we will understood the DNA image in next slide. So the membrane each base pairs can fit together with the double helix only if the 2 strands of a helix anti-parallel by the only the polarity of the one strand with the orientation opposite that of the other strand. So exact correspondence between the 4-letter nucleotides alphabets in DNA and the 20 amino acids are the present in a protein, that the genetic codes. So now we will move on to the another one small topic, DNA and ligand interaction. So the interaction of ligand with the double-stranded DNA is the fundamental, maybe is the intracellular process. Especially the protein that bind to specific DNA target sequence on the control of variety of process such as the regulation, transcription and translation. So suppose that protein will interact with DNA, this have a so many varieties of process. Now protein binds with DNA through the electrostatic interaction, either the salt bridge or the dipolar interaction, hydrogen bond, electrostatic or entropic effect, hydrophobic interaction or dispersion force like base stacking. So now easily you will understood what is nucleotide. So one of the phosphate group and these are the base and one of the sugar molecule. So this part will call as a nucleotide. So you can see this is the straight like a ladder kind of structure, sugar and phosphate backbone, and this is the base pairs. This is the double helix structure of DNA. Now you can see another one, actually what is the skeleton of the DNA structure. You can see this is the 5 ends and 3 ends. These are the hydrogen bonds. This is the phosphate group and this is the sugar group. This is the base pairs Thus guanine and cytosines interaction of red color indicate the hydrogen bond. This is the one strand and this is another one strand. This is the actual skeleton of the DNA structure. You can see here how the phosphate groups interact with the one phosphate group to another one phosphate group through sugar molecules. Okay, this is the actual structure of DNA molecules. Now you can see here how the DNA molecules now recording like minor groove and the major groove bindings and sugar phosphate backbone and adenine, thymine, guanine, cytosine. Now you can see any ligand will interact of this part, that is called major groove. Suppose any ligand will bind it, this part is called minor groove binding. Okay, this point is very important. Now we will see here actual what is the skeleton of the structure of DNA molecules. You can see here one strands to another one strand, what are the bonds, what is nucleotide, sugar phosphate backbone and hydrogen bonds, and phosphate, sugar, adenine, nitrogen containing base pairs. Now you can see what is the difference between DNA and RNA molecules. DNA is one of the double stranded and RNA is a single stranded nucleotides. Now you can see especially the RNA having uracils and DNA having adenine, guanine, cytosines and thymine. So this is the differentiation between DNA and RNA. Now you can see here when you record the spectra of UV spectroscopy like absorption spectroscopy, you can see find various wavelength and you can see relative absorbance, denaturing of the 82 degrees Celsius single stranded DNA. So single stranded DNA, it moves onto the hypochromic shift and hyperchromic shift. Okay, then you can see here UV absorption. You can see how when we move the various temperature from lower temperature to higher temperature, when we move to that above the 90 degrees Celsius, random coil will convert into the single stranded DNA. And then this is the DNA molecules and this is the RNA molecules. Now you can see how the DNA standards, hydrogen bonds and base pair bonds, and what is nucleotides, what is sugar molecules and what is the phosphate. You can see here one of the sugar molecules and this is the phosphate group and interaction with the base pairs, and this is the hydrogen bonds. This is called as a DNA strand. Now you can see clearly the schematic diagram representation, what is base pairs, what is base and what is nucleotides. You can see that by the sake of clarity we have marked in the figure or the basic concept we use as in the paper. The 4 types of bases: adenine, guanine, cytosine, thymine, 5 carbon sugar deoxyribose, blue pentanone. These are the how the DNA structure is looks like. Now we can see here what is dispersion, self-assemble. When the DNA will move the various factor applying on the DNA structure or DNA solution, it will be changed to various the aggregation, amorphous, normal DNA, fragmented DNA. You can see how DNA is denatured, and you can see DNA origami transmission, electron microscopy, various images we will recorded, and you can see DNA, how DNA looks like through the microscopy structure. Why is the normal DNA and aggregated DNA, dispersed DNA and fibril DNAs, crystal DNAs. You can see these the various transmission electron microscopy in the plasmid of DNA. And these are the some more images of the electron microscopy DNA structure. Now you can see here how the DNA is extremely unstable double stranded molecules that is prone to the breakage. How DNA is heat, suppose then it will goes to that repulsion kind of things, then heat, then will goes to the single stranded DNA. By cooling it will come to again double stranded DNA. Reverse transcription, you can see this forward reaction and backward reaction. And these are the how that bonds are happening. Hydrogen bond involving the nitrogen and oxygen. Nitrogen and oxygen, this is the hydrogen bond, that is the distance of the angstroms you are presenting here. So this is that class. Next class we will discuss very briefly and detail about the DNA structure Really, if you like this video, please like, share and comment. Thank you very much for your support, and specially heartful thanks for spending of your valuable time to listen this class. We will meet you in next class.